Good morning, good afternoon, and good evening, everyone. Before we get started, I'd like to acknowledge that I'm speaking to you from Treaty 6 territory in the homeland of the Métis. We pay our respects to the First Nation and Métis ancestors of this place, and we reaffirm our relationship with one another. And welcome to the 6th Annual Plant Phenotyping and Imaging Research Center, or PERC, Symposium. My name is Steve Webb, and I'm the Chief Executive Officer of the Global Institute for Food Security, or GIFS, here in Saskatoon, Saskatchewan at the University of Saskatchewan. And some of the background on GIFS, we're a public-private partnership that was established in 2012, and our mission is to work with partners to discover, develop, and deliver innovative solutions for globally sustainable food production. We serve as agriculture's innovation catalyst, connecting the agri-food ecosystem, advancing innovation, and bridging the gap to commercialization to deliver resilient and sustainable food security for all stakeholders. The PERC program is funded by Canada First Research Excellence Fund, or CFREF. And the CFREF awards are unique in Canada in that they are institutional awards and not awards for individual principal investigators. And the University of Saskatchewan is the only university in Canada that actually holds 2 of these awards. The PERC program, the symposium we're here for today and tomorrow, is managed by GIFS on behalf of the University of Saskatchewan. And our sister institute, the Global Institute for Water Security, manages the Global Water Futures CFREF program on behalf of the University of Saskatchewan. PERC delivers more than just outputs such as knowledge creation, high impact journal articles, symposiums like this, and the training of HQPs. There's an expectation with the CFREF programs to actually deliver commercial outcomes and impact. And that's one of the reasons why we at the Global Institute for Food Security have been asked to help manage this on behalf of the university. We've created, through the PERC program, our AgTech portal and hub, which is managed by the GIFS Director of Business Development, Chris Barker, as well as we've launched earlier this year the full commercialization of our Omics and Precision Agriculture Lab, which again uses outputs from the PERC program to be able to enable companies and researchers across Canada, the United States, and actually around the world benefit from the impact of the outcomes that this program and the scientists in it have delivered. Bit background on the symposium. We're excited to be able to put this event on for this year. We have well over 330 people have registered. In fact, it's still going up, and from over 15 different countries. Again, it's one of this year is one of the biggest events that we've ever had. And again, broader participation potentially through this format. We've got a great lineup of speakers. I'm going to put a plug in for Dr. Cranos Williams, our first keynote speaker in the first session. And I had the opportunity to meet Dr. Williams because I sit on the advisory board, the research advisory board of the College of Agriculture and Life Sciences at North Carolina State University. And Dr. Williams is leading a program that is very similar to what we're doing in PERC. And again, I think it's the benefit of working and building these international connections so that we can share, collaborate, and catalyze new innovations. We also have a panel from Nutrien that will be speaking this afternoon, and you could argue that this is a self-indulging plug for the panel that I'm going to be hosting. Again, it's 3:00 this afternoon, but again, Candice Lang, the Vice President for Sustainability and Stakeholder Engagement, Michelle Nutting, and Michael Nemeth are all going to be talking about carbon and for all the way from policy and regulation, the business model, and the practical challenges of implementing it with our most important partners, growers and producers. And I'd like to also thank Perform Media for helping us host this event. They are terrific partners. We value them a lot, and they've been a great organization to work with. Highly recommend them, and they've been pivotal to the last 2 symposia that we've conducted. I'd also like to thank our PERC Symposium Program Committee for all their work to help run this event. All the speakers that have that have prepared and are going to be present. And putting on a virtual event requires a great deal of coordination and planning. We're thankful for all that have made this possible. The other thing that I do want to mention is that it's also an interesting transition for us in the PERC program. Andy Sharpe has been the program director for the last 3 years of the PERC program, and Andy's going to be transitioning to a role, a bigger scientific leadership role as part of the GIFS Bangladesh collaboration. And in fact, he's the ideal candidate to help us run our major collaboration with the Bangladesh Ministry of Agriculture, in that he brings a wealth of experience from managing a broad multidisciplinary program such as PERC to a program that is even more complex with Bangladesh. Also, Andy's leveraging much of the outputs from the PERC program, including our Opal facility, to help basically nucleate and catalyze the efforts around our new Bangladesh initiative. So this will be Andy's last official event as the program director, but make it abundantly clear, Andy is still a PI involved with the PERC program. So he's not leaving us in that role. I want to also now acknowledge and thank Ian Stabness. Ian has been a key contributor and leader in the PERC program from its inception. He's served on the research committee, helped manage the transition from our themes to our flagships, and he's played a pivotal role on the program management team for almost the last 8 months as part of helping manage the seamless transition. Ian's entrepreneurial experience, and passion, I think, serve well as the program leader for the PERC program as we move forward. So again, I want to thank Andy. Please join me in congratulating Andy for his success as the program leader and congratulate him for his new opportunity. And I want to thank Ian, and please congratulate him for coming on to be the director of the PERC program. This is a pivotal program for the University of Saskatchewan, for the Global Institute for Food Security, but more importantly, it's a pivotal program for producers, farmers here in Canada and around the world. So with that, I'm going to hand this over to Andy, who is going to be facilitating our very first session. Andy, take it away.
Thank you, Steve. Thanks for the kind words, and welcome everyone to the 6th annual PERC Symposium. My time leading the PERC program has been extremely rewarding, and I'm really thankful to work with so many people doing such great work in the agriculture space, many of whom you'll be hearing from over the next 2 days as part of the symposium. I'd also like to acknowledge Ian and give him my best wishes. I really have enjoyed working with him over the last few years in the PERC, and I know the initiative will be in excellent hands under his leadership. As Steve mentioned, although I'm stepping down from the program director's role, I will still maintain a research interest, particularly in Flagship 1. So I'm really looking forward to the first session this morning, which I hope will demonstrate how omics, imaging technologies, and computational analysis can be utilized together First of all, I should give you information about what to expect during the session. The talks from our speakers this morning have all been pre-recorded and will play back to back, followed by a live question and answer session once they're all played, and I'll be playing a facilitating role in that session. Please direct any questions you may have to the chat on the right hand side. Once the question and answer session starts, I'll draw from these questions to spur discussion with our speakers. We have 5 speakers this morning. First of all, as Steve mentioned, we have Cranos Williams from North Carolina State University, and he'll be speaking about the application of machine learning approaches for modeling plant systems at different scales. Then we have Lingling Jin. She's a recent PERC-funded assistant professor faculty member at the University of Saskatchewan Department of Computer Science, and she's going to be talking about computational modeling of genome evolution. Then we have Mik Cieslak, He's a postdoctoral researcher from the University of Calgary, and he's been presenting on modeling plant architecture for image-based phenomics. Following him, we have Christopher Phoenix. He's an assistant professor in the Department of Chemistry at USask, and he's going to be presenting on PET imaging for monitoring ABA transport in plants. And finally, we have Venkat Bandi. He's a software developer at the USask Department of Computer Science. He's going to be presenting on interactive genome visualization tools. So with the introductions completed, we can begin the talks, and I'll see you again at the question and answer session. Thanks a lot.
I would first like to thank the conference organizers for inviting me to give this talk. My name is Dr. Cranos Williams. I'm a professor in the Electrical and Computer Engineering Department at North Carolina State University. I also serve as the platform director for the North Carolina Plant Sciences Initiative, specifically for the Data-Driven Plant Sciences platform. And today I will be giving a presentation entitled Computational Intelligence and Machine Learning Approaches for Modeling Plant Systems Within and Across Biological Scale. I currently lead the Embassys Research Lab, where our lab mission is focused on advancing the comprehensive understanding of emergent properties of biomolecular pathways associated with plant growth, development, and adaptation. And our goal is to do this by developing multi-scale systems modeling approaches. Overall, our group focuses on developing techniques and methodologies familiar to electrical engineering, such as machine learning, pattern recognition, and systems modeling, and use those methodologies to model biological processes across scale. The goal here is to be able to use these models to predict the impact of internal and external perturbation on plant systems. So this slide gives an overview of the types of things that we look at in the lab. We look at the ability to acquire and analyze data across biological scale, from the molecular and tissue levels all the way up to the organism and the extrinsic factors that influence plant development. We look at developing technologies that can analyze these data within and across scales to be able to generate models that would allow for new scientific knowledge, the generation of new research questions, and from an applied approach, improve decision support for stakeholders. So as part of the lab, we've looked at a variety of projects. And today I will be talking about two projects in particular. One focused on assessing how roots in Arabidopsis respond to single and combinatorial stress and the influence that has on root cell morphology. And I'll be talking about a project that's focused on the development of a multi-scale model for capturing the regulation and biosynthesis of lignin and how the biosynthesis of lignin impacts things like wood traits, specifically when lignin-specific genes are modified. So in this first project, the research question that we're going after is essentially what types of interactions exist between stresses that can impact plant morphology. The stresses that we look at in this particular project are iron deprivation and heat stress. And the idea is to assess how these stresses impact the plant by themselves and how they impact the plant under combinatorial conditions. So we know that, for example, heat stress impacts plant growth. It impairs photosynthesis, leading to reduced biomass. It reduces primary root growth and leads to the cytotoxic accumulation of reactive oxygen species. Additionally, iron deprivation also reduces primary root growth, increases lateral root growth, and impairs chlorophyll biosynthesis. Now, we all know that stresses in the real world do not typically occur in isolation. And the question that we really want to be able to assess is what is the resulting effect on the plant when these stresses are combined? And are there ways to decipher the interplay between these stress responses? So one way of doing this is to be able to identify a proxy for the influence of the overall stress. And through the use of this proxy and the quantified data that comes from it, we want to be able to access that proxy under different conditions. In this case, a control condition, a single heat stress condition, a single iron deprivation condition, and the condition of having both heat and iron deprivation applied. We then want to use this information and the quantified proxy to assess the differential effect of the single stresses and the combinatorial stresses based on the control. We then use this assessment to characterize the type of potential interaction for the combined stress. So for example, if the response of the combined stress is similar to what you would think you would see by combining the individual responses for the single stresses, then we classify the interaction as potentially additive. If the effect of the combinatorial stress is significantly more than what you would expect by combining the individual stresses, then we classify that as synergistic. And if the result of the influence of the combinatorial stress is less than what you would expect to see if you combine the two stresses, we characterize that as antagonistic. And the specific questions that we wanted to be able to address as part of this project is, can we develop computational approaches that would allow us to assess this through assessment and analysis of the quantified data from this stress proxy? Based on that, can we then quantify how this interaction changes over time? And can we use that knowledge of how it changes over time to then guide subsequent experiments for elucidating molecular mechanisms? So this work was done in collaboration with Dr. Ross Sudzani. Dr. Ross Sudzani generated or created what's called the magic growth chamber. This is a chamber that was built to serve as an addition to the light sheet microscope, the Zeiss light sheet microscope. We use light sheet microscopy to track the spatial and temporal expression of the fluorescently tagged genes in Arabidopsis root. In particular, light sheet was used to reduce photo-induced cellular toxicity and fluorophore bleaching. In terms of the experimental setup, as I stated before, we used the light sheet microscope. We used Arabidopsis thaliana marker lines where cyclin B11 was marked with GFP. And in this case, cyclin B11 is involved in cell division, and thus we postulated would give us a good approximation for the influence of the stress on root growth. We imaged the Arabidopsis roots under these four conditions: the control, iron deprivation condition, heat, and combined heat and iron deprivation, imaging the roots at 20-minute intervals over a 24-hour period. We collected both the fluorescent channel and the bright field channel. We then needed to create a piece of software to extract information from the images that were collected. We developed the bioevasion tracker, which allowed us to analyze various aspects, such as being able to track the root tips, being able to identify and segment the fluorescence associated with the expression of cyclin B11, being able to assess the spatial location of this expression with regards to the distance to the root tip, and with regards to a cylindrical coordinate system that describes the spatial location in the root, and developed a tracking system that allowed us to track this expression over time. So in doing this, we first looked at using the imaging software to quantify root growth and root growth rate for the 4 different conditions. And we did this over a 24-hour period. We then assessed the overall growth of all of these conditions over the entire 24 hours. And these results are shown here, where we see that for the single stresses, the overall root growth was significantly different once the single stress has been applied from the control. But we saw something interesting when we applied the combined condition. We saw that the overall root growth was not significantly different from the control. This begged the question of, well, if this is the overall phenotypic outcome, are we seeing similar types of characteristics at the molecular scale? To assess this, we looked at generating 10 metrics from the extracted characteristics of the light sheet images. In this case, these 10 metrics fell into 2 categories, spatial and temporal, where the spatial metrics describe the distribution of cyclin B11 expression using the mean and standard deviation from the distance from the tip and from the distance from the center of the root. The temporal metrics describe things like persistency, or how long the regions were expressed, or timing in terms of the timing in which they appeared, disappeared, or whether they were currently present. Using these metrics, we were able to develop time course signals associated with each of the 4 control conditions for all of the 10 metrics. We then wanted to try to figure out how to generate a proxy for the overall impact of these different stress conditions. To do this, we used the sum of squared distance reference based on any 2 experimental conditions, computed the sum of squared distance for all of the corresponding metrics, and then looked at generating the overall sum of square metric for any pairwise condition. So in this case, we see here curves that correspond to the response to iron deprivation with respect to the control, the response to heat with respect to the control, and a response to the combined condition with respect to the control. We then sum this over a 24-hour period to assess the corresponding differences. And what we see here is that we had an overall differential impact for the single iron deprivation stress. We had an overall impact of the heat stress, but when we looked at the combined effect with respect to the control, it was much less than what we would anticipate if we combined the 2 single stresses. This allowed us to infer that the response under combinatorial conditions was antagonistic. So in conclusion, we were able to assess some level of antagonism through an assessment of the root growth. This was further confirmed by looking at dissimilarity measurements associated with the spatial temporal metrics that we generated in terms of tracking cyclin B11 expression over a 24-hour period. These metrics shed light on exactly where this potential interaction could be occurring, where we see here the primary difference between the combinatorial conditions shown in yellow and, for example, the single condition in terms of the heat. The maximum differences occur over the 7-hour to about 15-hour time period. This provides a window upon which to guide subsequent RNA-seq experiments to then further identify or elucidate the molecular mechanisms that's causing this differential between the single stresses and the combinatorial stresses. The next project I want to talk about is a multi-scale modeling project that looked at developing a multi-scale model that combined transcript to enzyme interactions, the metabolic pathway, and how the results of the metabolic pathway could be used to predict phenotypic or wood traits. So this project started through initial collaborations with the Forest Biotechnology Group, where we wanted to formulate a multi-scale model of lignin biosynthesis to better understand how changes in specific monolignal genes impacted overall bioenergy traits. So in this case, the initial model looked at a very simple transcript model, where in this case, we wanted to predict the enzyme abundances based on the transcript abundance. And the assumption was that a given gene's enzyme abundance was only dependent on its transcript abundance. We then connected the resulting enzyme abundances to a differential equation model of lignin biosynthesis and looked at the steady state metabolites and fluxes as predictors of the bioenergy traits using a multilinear regression model. We wanted to explore potential improvements of this model by testing this assumption that the enzyme abundance was only dependent on its own transcript, because we know well that oftentimes that can be the case, but there are many situations where the enzyme abundance does not track the transcript abundance. So we wanted to develop a model to capture potential cross influences, likely indirect influences, that would help to contribute to the prediction of both transcript abundance and enzyme abundance. We then connected that to the metabolic flux model and replaced the multilinear regression model with a random forest model, because we wanted to assess whether there were additional nonlinearities that would help us to be able to describe or predict the wood traits based on steady state fluxes. So in this case, the data generated was data from wild type and transgenic trees, where 21 lignin-specific genes were knocked down or manipulated. This resulted in 207 transcriptome measurement profiles and 18 wild-type profiles, where we were able to capture both transcript abundance and enzyme abundance. And these were the data that were used to assess potential cross-influences between these components. We also captured phenotypic data, specifically bioenergy traits that essentially were categorized into several categories such as sugars, physical characteristics like height, diameter, and volume, and lignin characteristics such as lignin content and S2G ratio. So the first step of the model, we wanted to assess whether or not we could capture potential cross-influences between transcript abundance and protein abundances. The initial model, the model looked at being able to describe any transcript or any protein within this system, 20 transcripts and 20 possible proteins, and writing that as a linear combination of the other transcripts and the other proteins. Putting this into model form, we ended up with the structure here where we have a matrix B that describes the interactions and the rest of the model as we see here. We made certain assumptions such as the transcripts could not affect itself or a protein could not affect itself. And this resulted in a B matrix where the diagonal was 0. We also assumed that the B matrix was sparse. In making this assumption, we were able to then use a sparse maximum likelihood estimation approach using the data to estimate the interactions associated with this B matrix. And reformulating the equation that I had on the previous slide, this would then allow us, once we have this B matrix, to be able to set a knockdown, a desired knockdown abundance for any transcript within this system, and then be able to predict any abundance, estimate the abundance of any transcript or any protein in terms of the lignin biosynthesis transcripts and proteins. So the question we wanted to ask was whether or not the resulting model that we obtained that captured these interactions between transcripts and proteins, would that serve to better predict the wood traits than the original model that we had that assumed no interactions? And like I said before, we replaced the multilinear regression part of this multi-scale model with a random forest approach, which was trained based on the measured enzyme abundances and the metabolic flux model. So the results here show in black the results of the predictions for the bioenergy traits for the new transcript model, and in gray, the results of the prediction of the bioenergy traits for the old transcript model. And what we see here is that for 23 of the 25 lignin and wood traits, we actually obtained better metrics. We obtained a lower sum of squared error and a higher R-squared value for the new transcript model versus the old transcript model. We were also able to run in silico measurements by essentially looking at in silico knockdowns of a given enzyme family or given gene family, where here we see a in silico knockdowns of the CalD5H1 and 2 family, and actually assess the predictions of different bioenergy traits. And again, we see the new model in black and the old model in gray. And what we see is that the predictions from the new model actually better track the experimental data that we had from the original knockdown experiments. So with this new assessment and this overall multi-scale model, we can now look at in silico simulations of combinatorial knockdowns, essentially knockdowns that we did not have experiments for. And we can look at what would happen if we knock down combinations of enzyme families and the influence that that would have on bioenergy traits. This allows us to then do various levels of assessment, where, for example, if we are looking at knocking down the PAL and CoA-OMT family, we can look at now changes in different bioenergy traits. And this would allow us to identify potential regions of this knockdown and identify places where we can do things like reduce lignin content, while at the same time, not too much detrimentally affecting height and density. And we can look at various combinations of knockdown to explore the potential space and then use this information to help guide subsequent experiments. So in conclusion, we developed a multi-scale model combining omics measurements from transcriptome experiments with kinetic-based model of lignin biosynthesis. We were also able to identify that cross interactions estimated through a sparse maximum likelihood approach was able to better predict bioenergy traits associated with lignin-specific gene knockdowns. We also were able to combine everything together to look at possible combinations of in silico experiments that would then be used to guide subsequent knockdown experiments. And some of the categories that we were able to identify are currently being tested by Dr. Jack Wang in the Forest Biotechnology Department. So this ends my presentation, and I look forward to any questions that you may have.
Hello, everyone. My name is Lingling Jin. It's my great pleasure to present my computational biology research program under the Flagship 1 of the PERC project. Today, I'm going to talk about some of my work in computational modeling of genome evolution. So a bit about myself. I'm a new faculty member, joined the U of S in July last year. I got my degrees in computer science, specifically in bioinformatics from the U of S under Dr. Mik Cieslak. And then I did my postdoc at Agriculture Canada with Dr. Isabel Parkin. Then I got my first faculty position at Thompson Rivers University before moving back to Saskatchewan. Broadly speaking, my research interest is about computational modeling of biological systems to understand their interactions and evolution by using highly efficient computation. In the next few minutes, I'm going to show a few examples of what I am doing related to genome evolution and the connection to the PERC project. The first example is the modeling of transposable elements evolution. Transposable elements are repetitive DNA sequences that can move themselves to new positions within the genome. They are highly abundant and active in plant genomes, contributing to plant phenotypes. For example, they have contributed to fruit shape and color of tomatoes. They also create genetic diversity and act as an environment responsive element for drought resistance in many plants. By modeling this nested insertion patterns of transposable elements in a single genome, we can use a tree to capture the evolutionary relationship of TEs. And then a TE network can be created by analyzing all the TE-rich region in the genome. This network represents the phylogeny of TEs in a whole genome scale. If we zoom in the network, you can see that TEs are aligned with their age groups, and their interactions are captured by the direction of edges. Lifespans are represented by the size of the nodes. By embedding genes of certain functions into the TE network, we will be able to examine how the proximity and activity of TEs regulates the expression of these genes. And another example of my research is about the modeling of genome rearrangements. By predicting the optimal rearrangement scenarios between the 3 brassica diploids, we constructed the ancestral genome architecture of brassica. And this work can reveal how gene contents and their ordering retained or changed within the brassica group. So note that canola is the hybrid of these 2 genomes. As canola being a member of the brassica group, the study about brassica ancestor improves our understanding about genomic stability and diversity of the canola crops. And in light of the study based on brassica model, we published a tool called RACROCH to reconstruct ancestral context and chromosomes in a given phylogenetic tree. For each ancestor in the tree, the pipeline can automatically cluster ancestral gene families into paleochromosomes. The diagram shows that RACROCH reconstructed 4 ancestor nodes from 6 orders in the monocot clade, where each branch in the tree has at least one whole genome duplication within that order. The projection of gene families between extant and ancestral genome shed light on how whole genome duplications shape the extant genome structure and provide the genetic raw material for evolutionary innovation. And note that wheat and rice, they both belong to the Poales order, and the ancestor genome of the monocot clade can improve our understanding about the modern wheat or rice genomes. In another project, we reconstructed ancestral genome structure at 3 key phylogenetic nodes of eudicot diversification. The core eudicots, represented by vitis or the grape genome in the diagram, descended from an ancient whole genome triplication called gamma in the eudicot clade and account for 70% of the present flowering plants. A floral feature called whorled phytonary with flower parts arranged in concentrate whorls of 5 actually evolved shortly after the origin of the core eudicots, and could be genetically linked to the ancient whole genome triplication event through multiplication and rearrangements of floral transcriptional regulations. And this work indicates that genome structure remained relatively stable during early eudicot diversification and acknowledges the role of gene and genome duplication, providing genetic raw material for evolutionary innovation. And then we also explored several orders in the Roset class in core eudicots to examine the diversity of plants while maintaining key functions from an evolutionary point of view. And canola and lentils, they both belong to the Roset class. So through computational modeling of genome evolution, including transposable elements, we can answer many important questions, such as what are the functions of ancestral genome and how they retain or evolve? What does ancient polyploid shape the extant genomes? And how about polyploidy, how they impact the expression of resistant genes? How transposable elements drive the evolution of resistant genes or contributed to various agronomically important phenotypes or response to different environment. And what is the monoploid status of ancestors and how can they contribute to the diversity of cultivated crops, such as the canola crops? So besides the genome evolution modeling, I also have a few other topics, such as leveraging machine learning to advance genome-wide association studies, metagenome or metabolome-wide association studies to evaluate the effects of crop rotation for wheat fusarium resistance, or to improve structural variance in identification with both short read or long read sequencing data. And also some ongoing project such as the synchrotron-based X-ray to determine fusarium infection, or greenhouse imaging and analytics for omics innovation in agriculture. That's about everything for today, and thanks for listening.
Hi, I'm Mik Cieslak. I'm a research associate in the Department of Computer Science at the University of Calgary, but I'm also part of the Plant Phenotyping and Imaging Research Center in Saskatoon. Over the last decade, a number of automated image-based methods have been developed to help extract plant traits in phenomic studies, including drones in the field. This has led to vast amounts of data, and although getting these images and data is complicated, interpreting them is a bigger challenge. For example, in screening a large number of plants for desirable traits. For this talk, I'm focusing on shoot phenotyping of canola plants grown in controlled environments, for which we have a large data set of plants grown in a greenhouse at a LambdaTech high-throughput phenotyping facility. These 8 images show how some of the data looks like for a single plant, but the entire data set contains 300 plants from 50 Brassicanapus genotypes, and we observed a large phenotypic variation in shoot architecture, including plant height and branching angle, plant posture, and physiological age. These are simple observations that can be made just by looking at the images, but are also examples of traits that image processing pipelines are not designed to extract, because they are not functions of the image pixels but are more semantic, like organ count, a plant's physiological age, if a plant is stressed, or a plant's potential yield. These traits normally have to be identified by a human expert, but recently machine learning has been applied to plant phenotyping to automate the process. In this contrived example, a neural network breaks down an input image into nodes and assesses what is in the image. Here the task is to classify if the plant is healthy or stressed. Initially, the weights are arbitrary, but after feeding in images with a known answer, the weights are adjusted so that the network returns with a high propensity the known answer. As an example, I've set weights in the hidden layer of nodes to show how the network might arrive at an answer. In practice, however, neural networks require large amounts of annotated data for training, which are time-consuming and expensive to obtain and are relatively small compared to the size of datasets often used for training them. One common approach to data augmentation, which has been shown to be effective, is to apply transformations like flipping, cropping, and distortions. This augmentation, however, is still limited by the size of the original dataset. We showed that images of synthetic plants can also be used to generate training data for a leaf counting task, and the neural network performed better at this task when we augmented the training with synthetic datasets. The model network, however, was a simple L-system-based model of an Arabidopsis rosette and was a proof of concept. Now we apply the same idea to canola. We constructed a developmental L-system-based model to simulate growth of a plant from seeding to rosette, bolting, flowering, and potting stages. The sequence here is for one generic plant, but we proposed a method for fast visual calibration of the model to images like those from a LambdaTech dataset. So now I'll show a demo of the calibration process. Calibration is performed in the context of multiple views of a single plant. In particular, a front view, a side view, and a top view, as well as a combined view in 3D. Calibration is initialized by dividing the main stem into vegetative and reproductive sections. Yellow shows the vegetative section, and blue the reproductive section. A slider is used to change the size of the sections to better match the image. Higher order branches can be enabled, and the user can control the distribution of lengths. And the branching angles. The user can then enable visualization of leaves and calibrate their sizes. as well as leaf inclination angles. Flowers and pods can also be enabled and calibrated in a similar way. Now, if the background images are suppressed, the final result is a calibrated model. This is a calibration for one phenotype, but we can apply the same technique for other phenotypes. This one has short branches with broad branching angles, whereas this other phenotype has long branches with narrow branching angles. Finally, the third phenotype has mostly straight branches with many pods. For the same phenotype, we can also calibrate the model to changes in growing conditions, for example, controlled versus water-stressed. The calibration can also be performed for multiple stages of a canola plant's development by setting the parameters that affect timing of development, like elongation rates of internodes or the rate of appearance of new organs. So far, I focused on isolated plants, but the model can also be used to simulate field-grown canola from early stages of growth to later stages. However, simulating many plants growing together requires including different aspects controlling the plant's development. For this, interactions between plants have to be considered, including the light environment, like the red to far red ratio, and mechanical interactions. Then, different synthetic phenotypes can be used to help study the architecture of the canopy by examining the spatial and temporal organization of the organs, the timing of flowering and the control of branching, and their effects on plant growth. In summary, the focus of our work is on generating annotated data for training neural networks in image-based phenotyping tasks, specifically for identifying semantic traits in images. To this end, we've developed a method for the visual calibration of models to images using L systems with time-varying positional information, which is a fast and effective way to generate such ground truth data. The next step in this work is to apply the images of synthetic plants to train a neural network on semantic traits. Nazifa Khan has a poster at this symposium showing our progress. Thank you for your time.
Okay, so good morning, everybody. My name is Chris Phoenix, and I'm going to tell you about our efforts to develop a PET radio tracer where we can image ABA transport in living plants. Now, full disclosure, my background is in biomedical research. Okay, so normally I'm developing radio tracers for humans and do a lot of rodent imaging. So this is really the first time I've done any kind of plant research. So forgive me if I use the wrong jargon or something like that. Now, PET imaging stands for positron emission tomography. This is a really powerful modality that's been used extensively in biomedical research, including guiding drug development. And it's already about a $2 billion industry in the US. It hasn't been used in plants since I don't have enough time to go into it, but it hasn't been used as extensively in plants as it has for human and animal research. Now, everybody in the audience here is familiar with abscisic acid, ABA, and what we tried to do is develop a F18 labeled radio tracer of ABA. And we would love to be able to use PET imaging as a non-invasive tool to understand ABA biology. For example, imaging long-distance transport of ABA in living plants, looking at metabolism of ABA, spatial regulation in different plant tissues, look at distribution and redistribution. And this is something that I think is really important, is use PET imaging as a tool to help develop new agrochemicals. And then, of course, characterizing ABA transport and flow kinetics in different plant phenotypes. Now, the advantages of using PET imaging is that it's non-invasive, it's non-destructive. We can image dynamic processes in living plants, such as the flow of ABA. It's applicable to all plants. We don't need to do any transgenics or genetic modifications. We just have the plant and do the imaging experiment. PET imaging is the most sensitive molecular imaging modality. Okay, so we use actually sub-pharmacological doses of the radio tracer in plants, animals, and humans. And this would be really important, for example, studying hormones in plants, is that you're using such a low dose that it's not going to have pharmacological properties. It's not going to influence the plant biology. And then you can pair PET imaging with autoradiography, metabolism studies, and ex vivo gamma counting, get an overall really accurate picture of ABA biology. Now, as a radiochemist, I often warn people, radiochemistry is very hard. Okay, it's notoriously challenging, and this is because we're using nanogram levels of the isotope. F18 is the isotope of choice for PET imaging, and it's produced in a cyclotron, but it has a very short half-life of only 2 hours. So we have to synthesize our radio tracer, purify it, and determine the structure using HPLC within usually a couple of hours. So that's really, racing the clock is a real big issue. And we can't characterize the radio tracer using standard organic chemistry techniques. But I'm very pleased to say that we were able to synthesize this radio tracer using this epoxide as a precursor in sufficient yields for lots of imaging studies. Okay, so once we had fluoro-ABA, we wanted to determine the catabolism of fluoro-ABA in canola. And is it similar to ABA? PET imaging doesn't see the molecule, it sees the isotope. So the catabolism was really important to sort out. So Sue's group synthesized various deuterated analogs of ABA as well as deuterated versions of the 3' fluoro-ABA. And we sent those to EG in Toronto to do mass spectrometry studies. And what he determined is that 3-fluoro-ABA is catabolized in a very similar manner to ABA. So that's good. And then to follow that experiment up, we fed our radio tracer to canola. We extracted the tissues and ran a radioactive HPLC, and we determined that after 3 hours, 80% of our radio tracer is still intact, 3' fluoro-ABA. And as expected, our major catabolite was fluorophthalic acid. We had 2 other minor catabolites, which we aren't really sure what those are at the moment, but we're trying to figure that out. So here's our preliminary, some of our first autoradiography images. We've done studies in Arabidopsis where we see ABA flowing down into the plant. Notably, we added the radio tracer. We punctured little holes in the leaf, and then it entered into the plant and was distributed. Here's some canola seedlings, and here's a larger plant. So this is a really neat image of a large canola leaf where the ABA was administered right there, and you can see the plant vasculature. And as we optimize our autoradiography method, the autoradiography images got better and better. Okay, so what kinds of things can we do with this now? Well, we can study different canola plants. So NAM0 versus NAM5 versus NAM1. NAM5 is drought sensitive, NAM1 is drought resistant. So here's an example of where we added the radio tracer to a solution and dipped the roots of the canola into the radioactive solution, and the ABA is transported up into the plant. And under drought conditions, we don't see as much radio tracer uptake, and this would make sense for the xylem transport in the drought, reduced xylem transport under drought conditions. Here's another experiment where we added it to the leaf, and we saw radio tracer distribution down into the roots. However, under drought conditions, we didn't see any distribution into the roots. So these are the kinds of things that we can look at. We also did a preliminary experiment where we looked at the radio tracer in the sap of NAM0, NAM1, and NAM5 canola lines, and we saw differences between the drought and the well-watered conditions. Now, this is just preliminary, so we have to repeat these experiments to really get a good idea of what's going on there. Now, so far, as I said, we want to develop a PET imaging method. So far, we've been using a PET scanner dedicated to animals, to rodents. And here's a PET scanner that is currently being developed, dedicated to plants. Now, here's our first PET images that we took of 3' fluoro-ABA distributing in a living canola plant. So it's very, very exciting stuff. And here's the CT scan so we can look at the plant anatomy. So that's all I'm showing you today. So thank you to my team, Sue and EG, Raju, really great collaborators. Thank you to all of our trainees who work very hard on this project and to the funders. And if anybody has any questions, feel free to contact anybody on the team.
The work I'll be presenting today is centered around a collection of visualization systems that were created under the supervision of Dr. Carl Gutwin at the HCI lab at the University of Saskatchewan. So the central idea behind any sort of visualization system is its ability to rely on human capacity for visual perception to support some form of cognitive processing. Or in much simpler terms, it's using visual aids to hold complex thoughts and process them. Now, in our particular context, we primarily focus on visualization systems that are centered around genomic datasets. One of the defining characteristics of genomic research in the last couple of decades has been the improvement in next-generation sequencing technologies that have reduced the cost of sequencing. This, of course, has led to the generation of enormous amounts of biological data, both about humans ourselves and the organisms that surround us. This, in turn, has led to a surge in demand for tools that researchers can rely on to make sense of this ever-increasing trove of data. And that's where visualization systems come in. Since even though a large part of genomic analysis is purely computational and can be automated, human judgment is still vital in certain scenarios. As humans, we're intuitively good at identifying patterns in spatial representations. And so biologists are increasingly relying on visualization analysis tools to both hypothesize and investigate diverse genomic tasks. However, one of the biggest impediments to using existing visualization tools is that they do not support a wide variety of investigative tasks and can often have limited or no interactivity at all and purely exist as chart generation systems for research publications. Thus, to address this gap, we set out to build a suite of visualization tools which are tailored to meet the demands of genome researchers. To do this, we met with different groups of genomic scientists and biologists at the University of Saskatchewan, and then curated the needs into different design requirements. One of our first tools that came out as part of this research is Synvisio. Synvisio is a web-based tool that's designed to assist researchers and biologists in interactively exploring genomic conservation. It offers multiple visual representations such as parallel plots, matrix-style dot plots, and even multi-comparison plots such as hive plots, which is the one that you can see on the bottom right. It uses a combination of color and spatial representations to visualize conservation through these colored ribbons that then link the conserved regions. Additionally, it can also be extended to compare several genomes at once through a tree plot, where we stack parallel plots vertically and then link the duplicated regions between them. In this case, we have lentils at the top, barrel medic in between, and then chickpea at the bottom, and all the conserved regions are linked between each other. So you can sort of trace the conservation from the top to the bottom. Apart from this, we've also built a web-based tool for haplotype maps that can be used to visualize differences in genotypes for molecular marker design. For this, we use datasets given by Dr. Kirsten Vett from Nopals and Dr. Isabel Parkin. So in this visualization at the top, we have the entire genome of lentils with one chromosome in each vertical zone, and then one germplasm in each horizontal line. We then assign a unique color for each germplasm, as you can see in the expanded view for chromosome 7, and then compare all of its SNP markers with the lines above it. So if a SNP matches any of the lines above it, it is assigned the color of the first matching line above. And then if not, it's given the unique color of the reference line. This helps in sort of creating patterns where certain SNP markers that get clustered around and conserved across all the lines. Users are also given the option to sort of interactively explore the visualization at different genomic scales. So they can start with the genome level and then can explore the data at the level of a chromosome or a particular region within a chromosome, such as a QTL region. We also have additional markers to enhance the visualization, such as copy number variations, which are these red and white dots that you can see in the system, and then gene annotations corresponding to the selected region, which are the blue arrows in the bottom. So additionally, if you can see it, you also have a small whisker plot attached to these copy number variation markers, which shows the extent of that marker. And for the gene annotations, you can sort of click on any of those blue arrows that you see in the bottom, and then you'll get a tooltip that tells you the function and additional notes that are attached to that particular gene. Now, one of the limitations of this system is that we can only compare 10 lines at a time, and that's inherently due to the number of limited colors that we have in the system. And so we built an extension to this system that can visualize large-scale patterns in slightly bigger data sets. So in this type of visualization, we have a germplasm line in canola being compared against 50 different lines in Brassica napus, and every SNP that matches the original line is colored in blue and mismatches are colored in red. As you can see, patterns start to emerge in the visualizations based on the clustering of the lines themselves. So the lines can be ordered either manually or they can be ordered based on a phylogenetic tree, as you can see on the left, to cluster similar lines together. Apart from this, you can also order the lines based on certain phenotypic traits like prime or similar such trait markers. In this particular case, we have a trait map to the left and the lines are simply ordered based on their origin. So we have all the Australian lines clustered to the top, and then the Canadian lines, the European, and then the exotic ones. This tool, just like the others, is also interactive, and users can sort of visualize the data at different genomic resolutions. So they can start at the genome level at the top and then drill all the way down to a particular QTL region. At the smallest resolution, you can see additional information such as SNP IDs and the alleles corresponding to each of these SNPs. And also users are given an option to view the gene annotations corresponding to that particular region and then look up the nodes and functions corresponding to those genes. Of course, due to the lack of time, I won't be able to demo all the other tools that we have built as part of our research, but please do check out our website, genomevist.usask.ca, where you can find direct links to all these tools that we have and some other new ones. And you can, of course, play around and explore them, as these are all fully web applications. Finally, in case I forgot to mention, these systems are all free and open sourced web apps, and so they can be hosted on your own servers with your own datasets if needed. Additionally, if you have any datasets that you'd like visualized either in these systems or other novel visualization systems, please do drop us a mail and we'd be happy to help you out. Thank you.
Well, hello everyone again. So now we can begin our question and answer session. Thanks to all the speakers. Some great talks there. And we have all the speakers apart from Lingling Jin, who unfortunately couldn't make this time slot because of a previous commitment. But anyway, we can begin with the questions. I'm going to be I can ask some questions, but I know there's some questions that come through on the feed as well, which is great. We'll begin with some questions to Cranos, and I'll go sequentially through the different speakers, and then hopefully there'll be a question or possibly multiple questions that could be asked to all the panelists. So to begin with, some questions to Cranos, and I'll start from the chat, Cranos. So there's actually a question from Leon Cochin at GIFS, and basically noting some interesting findings on the heat and iron deficiency stress. He has some questions. So why did you choose these two stresses in this combinatorial study? Basically, it's not being critical, but he reckons he's not often seen these two stresses studied together, although, of course, they can occur in the real world. So can you give a bit of a rationale as to why you chose those two stresses?
Yes. Thank you for your question, Leon. We did take into consideration the likely co-occurrence of these given stresses in the real world as a mechanism for trying to convey relevance of the findings that we had. Also, individually, the collaborators that I have in the plant and microbial biology department had studied these stresses individually. So we have individuals that have not only run experiments on these stresses, but are experts in the responses associated with both iron deprivation and heat response. So what we really wanted to see or assess was whether or not these stresses, given that they have very similar effects, both molecularly and physiologically, would the effect, in a sense, compound on itself or be exacerbated when we looked at them together. And this is what we were able to assess through our analysis.
Okay, thanks for that explanation. I guess I have a bit of a follow-up. And I guess it's related to potentially other stresses that you might be looking at. So water stress, for example, as another abiotic stress, but also biotic stresses. Have you considered some other stresses? And I guess also, can you reflect perhaps on how far this combinatorial approach could go? How many different stresses can you compound?
So that's a very good question, Andy. And I think it really comes down to the mechanisms of the experimental setup and obviously the limitations thereof. With the iron deprivation and heat stress, it really was amenable for the setup that we had for the light sheet microscope. Obviously, if you start to look at other types of stresses, whether it be water stress, or in this case, drought stress, or other types of biotic stresses, ensuring that, we have the right experimental setup is going to be key. And then, of course, as you combine the different, expand the different combinations, it becomes more and more prohibitive. So I think as we move forward with this type of study, being able to highlight things that potentially, like you said, occur in the real world, or things that could potentially be translated to more field type concerns or crops that will be grown in the field, I think that's where the direction of this type of combinatorial assessment of stresses would go.
Okay, thanks. So there's another question from Leon with respect to how in the first study you present how the Arabidopsis roots were grown. Can the light sheet microscopy allow you to image fluorescently labeled roots in the soil?
So in this particular case, the setup that was created by Dr. Ross Sidzani, essentially the roots are grown in a clear caliper, similar to what is normally used for the light sheet, but it was constructed in a way that allowed us to image in rotation 4 different roots at a time. Now, this clear caliper, the roots were essentially grown within this caliper and in a clear media. So in this particular setup, we would not be able to look at the fluorescence in something like soil because it just wouldn't work in the light sheet.
And finally, the final question from Leon is, did these stresses preferentially inhibit cell division or cell elongation in the root growth zone, or were both inhibited relatively similarly?
So that's a very good question. So we really did not look at the individual developmental zones of the root independently. We only looked at the entire root as a whole. Now, of course, follow-up experiments would be helpful, specifically in terms of isolating the given developmental. In terms of looking at the individual developmental zones, that is a likely follow-up, a possible follow-up. Now, how to go about doing that would be key. We could look at the fluorescence of cyclin B11 in the different developmental zones. Of course, this would likely require another additional marker line that would either help us to localize the different developmental zones based on cell characteristics, for example, size of the cortex or size of the various outer cells of the root that are indicators of the different developmental zones, or look at specific marker lines that are indicating, that would help us to indicate the specific developmental zones. With regards to the overall effect, the effects and the analysis that we had was really focused on cell division. So, using cyclin B11 as a proxy for cell division, and then, of course, looking at the whole root, that's what we were able to assess. But I do agree that further looking at the different developmental zones, and in particular, potentially even expanding this to looking at, say, something like RNA-seq, over the time range that we were able to highlight in the results between 7 and 15 hours. That would also help to provide insight into the potential mechanisms that could be affecting the different developmental zones as well.
Right, so I guess I got a bit of a follow-up question there. You talked about the spatial and temporal metrics and using that as a guide to when you do your transcriptional profiling, your RNA-seq. So in order to carry out the RNA-seq work, are you actually going to be looking at particular tissues within the roots? Are you going to be using laser dissection for pulling out those tissues and getting specific RNA-seq libraries, and that over I guess a temporal range?
That is an excellent question. So we've had the ability to do somewhat all of the above, or at least in the past, have done all of the above in some of the experimental work that my collaborators have done. We've either looked at whole root transcriptomics. We've also used things like FACS to isolate, for example, epidermis cells. We've also looked at exploring, for example, the development of gene regulatory networks in different developmental zones. So really, it kind of boils down to what the next logical question is. So in the case of, for example, Leon, if the focus was to really ask the question whether or not the influence on changes in overall root length is really associated with specific molecular mechanisms in the different developmental zones, then isolating the developmental zones would be key. If there was reason to believe that there were specific cell types that really were contributing to this potential synergistic type of effect due to the combinatorial stress, then we could go about trying to isolate the specific cells. That probably would not be the first route that I would go, because doing the specific cell type separation we've done it before, and it can be pretty time consuming, especially to be able to get enough material to be able to run RNA-seq. So looking at the developmental zones, for example, would be a good first choice. Or, for example, looking at the whole root and then assessing, what types of differences in differential expression we see across the single and combinatorial stress conditions.
Okay, thanks. There is another question from Stanley Adebar at USask, and it's kind of going back to that temporal study in the Arabidopsis roots. So do you have data and results beyond 24 hours? And if you do, how are the results beyond 24 hours compared to the 24-hour period?
Okay, so the answer to that question is no. We do not have results after 24 hours. And to some extent, we were hesitant to go beyond 24 hours because, again, overall, especially when you start looking at long-term growth experiments in something like the light sheet, especially as the root continues to grow beyond, for example, the potential length of the capillary, the plants themselves can get somewhat stressed. We would likely need to look at potentially designing a different apparatus to be able to extend this beyond 24 hours. So looking at, let's say, 24, 36, 48, maybe even up to 72 hours. But I think that's something that would be interesting to see, whether or not we see either cyclic types of characteristics that are potentially influenced by the circadian rhythms, or we see some level of an exacerbation of the overall effect that we're seeing due to the single and combinatorial stresses. But that's something that we would have to look at in the future, and we would likely have to redesign the experimental setup. Okay, thanks.
I have a quick question regarding the second part of the talk on the lignin biosynthesis. So in theory, the model could be implemented across any biosynthetic pathway, I assume. And are you actually looking at other pathways in plants or other systems?
With regards to this particular work, no, not yet. And primarily the rationale isn't because of the potential applicability of the approach itself, but due to the amount of resources and experiments that would need to be run to be able to reconstruct something similar for another developmental pathway. I think, when I mentioned the experimental data, the context in terms of time that it took to acquire all of that information was relatively painstaking. That took probably a project over about 5 to 7 years. So there were many different types of in vitro kinetics that were ran for all of the enzymes that were associated with the lignin biosynthesis pathway. All of those things went into the development of the parameters for the metabolic pathway. All of the, again, experiments in terms of the transcriptomics, the enzyme abundances, so in theory, yes, it likely could be done, the same type of approach, but we would have to have similar types of resources to be able to have all of the data that would be necessary. So right now, what we're really focused on is more so the validation and verification of the current multi-scale model. So as I mentioned in the presentation, we're currently collaborating with Jack Wang in the Forest Biotechnology Department to look at how we can use CRISPR to do some of these potentially targeted combinatorial knockdowns and then test the results of the combinatorial in silico simulations from the model.
Yeah, I assume one factor might also be when you're working with trees, the life cycle period is long, right? So in other plant systems, it could be a bit quicker with a shorter life cycle.
You're absolutely right. Yeah, I was astounded to see how long some of that took. Not only that, but the space that it requires to grow that many trees was tremendous.
Okay. Well, thanks, Cranos. I'll move on to some questions for other speakers now, but I'll be coming back to Cranos for a more general question. Mik, there is a question in the chat feed, and it's from Jordan Williams, who I think you know, at the University of Saskatchewan. Mik, the simulator looks amazing. Can the simulator sample randomly from the parameter space, like in our methods paper, or is each model tuned by hand?
Yes, hi, thanks. So hi, Jordan. Thanks for the question and the comment on the simulator. So yes, we basically have built upon that methods paper for randomly sampling from different distributions at least for simple models of vegetative growth, like in the Arabidopsis rosette that we did before, and currently we're doing the same thing for the maize. Well, anyway, the vegetative growth of maize plants. But for canola, it's a little bit more complicated because, as we showed in that paper, you need to generate a distribution of traits for the training data set for it to actually work pretty well. So for the canola paper, we're planning on, it's been hand-tuned to several different phenotypes, let's say. And then we're planning on using those 3 or 4 hand-tuned models to generate the parameter space. And when you have a parameter space, then the model can sample from that parameter space. So it should be kind of the next step of what we presented in that methods paper. And something like that has already been done for, say, modeling architecture of trees. So you generate just one model with many different parameters. You tune it to some of the known or some of the known architectures, and you can generate things in between. Or even inflorescences. There's papers on generating different types of inflorescences in the same way.
Yeah, thanks, Mik. I've got a follow-up question. So you mentioned you're working with the NAM parental line, so the Brassicanophis, the canola nest association mapping population, which is, I guess, one of the core germplasm resources for the PERC initiative. It's controlled environment data, and I think there's data available for all 50 lines. Have you actually carried out those fast calibration model work and simulations with the data from all 50 lines, or is that in progress at the moment?
Yes, actually, that's very related to Jordan's question. So instead of fine-tuning to all 50 of the plants, we're planning on just generate some of them and or fine-tune the model to some of the kind of, let's say, more distinct architectures, and then use the model to generate everything in between without having to hand-tune it. That's the plan anyway. I'm not quite sure if it's going to work yet, but I hope so.
Yeah, I mean, you have the data as well. You just have data from the parental man lines. And another thing that could be a follow-up is actually going to the individual subpopulations in the population itself, because I think it's about 2,500 lines in total, and as 50 lines from each cross of the NAM donor line with the reference line. So you'd actually have the ability to look at data from individual populations or subsets of populations as well.
It'd be interesting. Sorry.
Yeah. Yes, it would. I mean, I'd have to get that data. We don't have it at the moment, but the other data that we do have is data from the field. So the NAM population has been grown out in the field over multiple years. And one thing I think you perhaps envision doing is comparing some of the data that we got from the controlled environment and your simulations with how the plants actually grow, and it's not individual plants, they're in plots. So you could actually maybe carry out a comparison to some of your simulations of field-grown plants with actual plot data.
Yes, for sure. Those kind of studies have been done, not for Brassica napus, but other brassicas, in fields testing how well they react to crowding or disease prevention, and simulations of different kind of spacing in the plot. The model, though, however, this model I presented is mostly descriptive. It doesn't include any kind of functional aspects of how plants grow. So branching angles might change if you crowd the plants too close together, or if you decapitate the plant, it's going to react. Whereas the current version of this model will not react. However, those kind of physiological aspects of growth have been modeled before and have been included in different types of models. So it is something that is certainly possible to do.
Okay, and the last question I have is, have you worked with other crops other than canola? Are you starting to extend things like wheat or perhaps other crops?
So the maize for this particular work we've done for maize and canola. In the past we've done plenty of different crops, but like, for example, kiwi fruit, but that's quite different from a cropland. It's a vine, yes.
Right.
Yeah. So we've been looking mostly at that, and the next focus, well, actually, a large focus of our work has been on Brassica oleracea, because we're interested in how such a variety of morphotypes can be generated within a single species, right? There's the broccoli, cauliflower, Brussels sprouts, cabbage, and they all look very different, yet they're the same species. So is it possible to make a model that would generate, just by changing a few parameters, could we explain how those morphotypes could be generated?
Right, yeah, it's known as the dog of the plant world.
Yeah.
Well, so very similar genome, but there's so many different morphotypes. Of course, vegetable types in oleracea. I guess it's largely perhaps controlled by a relatively small number of genes, or variation in a smaller number of genes that are affecting the architecture. So modeling that would be tractable, I think. Yeah.
Yeah. Even for Brassica napus, I believe we've been working with some researchers from the John Innes Center. They have a large collection of of a diversity of plants as well, and they're interested in using these methods for the same thing we're trying to do at PERC.
Yeah, that's true. Canola has different types. There's spring types, there's winter types, of course. Winter types generally grown in Europe, and spring types in North America. But then there's also the vegetable types as well, the sweets. So similar to a bit of similarity with the oleracea morphotypes. Okay, thanks. Thanks, Mik. I'll move on to Chris now. And I think there's a question to you, Chris. Yeah, from Nia Vade from the University of Calgary. Has the sensitivity and the accuracy of the ABA probe been compared to other available fluorescent FRET-based reporter systems? Also, considering the short half-life of the probe, how long can ABA be tracked in the living system, I guess, practically?
Yeah, thanks for the question, Nia. That's a very good point. I think I haven't compared the sensitivity of the radio tracer to the fluorescent techniques, which I assume you're talking about, like ABA Leons for detecting ABA in plants. But one of the strengths of the PET technique is its sensitivity. So we're actually administering, nanogram levels in total to the plant. and PET can detect picomolar concentrations of the radio tracer. So I think that we're, well beyond, not well beyond the sensitivity of fluorescence, but I think we're have higher sensitivity. And the thing that I think is important to consider is that some of the fluorescent proteins that they use act as a sink for binding ABA, and so it can actually blunt response. Whereas for the PET tracer, we're really at tracer levels, and therefore it can detect flow around a plant without perturbing the biology of the plant at all. So I think that's one of the strengths. And one of the weaknesses, like you said, is fluorine-18 has a 2-hour half-life. So that's great for injecting radio tracers into humans due to the lower radiation exposure, but that limits our imaging experiments to about 4 hours top. So sort of longer term studies can't be done with F18. Of course, there's dozens of other PET isotopes we could use, but now you're talking about radiometals, which are bigger than the ABA molecule itself, so that wouldn't be very useful. So that is a limitation for sure, is that we can't do imaging experiments probably beyond 3 to 4 hour ranges.
Right. So I guess with your setup, you actually have a lab in the cyclotron building, so you can generate the isotope and then use it effectively immediately. So I guess if you have that capacity of locally available, you can do the work.
Yeah, and that's one of the things that we've really worked hard to do here at Saskatchewan is set up the radiochemistry facilities, the imaging facilities, both for plant and animal. It's literally down the hallway. So when I was a postdoc in Vancouver at UBC and Triumph, when we had to do an animal study, I had to literally coordinate with 3 different organizations to get that experiment set up. Whereas here at the cyclotron, Saskatchewan Center for Cyclotron Sciences, we do the radiochemistry, we validate that we got the right tracer. Down the hall is a plant growth chamber, and the PET system literally feet away from each other. So we just walk over and do the imaging experiment. So it's really set up nicely. So now we can do different types of growth conditions, different NAM0, NAM1, NAM5 kind of different strains of canola, and look at those. In an afternoon, we can do 3 or 4 different PET imaging studies.
Mm. Yeah, I must admit I was impressed with well, I guess back a year or 2 years, whenever we had the tour, with the facilities that have been developed for the plant work, the growth cabinets, and it looked like a nice infrastructure to utilize for the work. I have another question. I guess it's going back to the NAM lines. You mentioned a couple of the NAM parental lines, and it's the same population, the same parental lines from the same population that we talked about with Mik. But have you just focused in on a small number of lines, or are you actually planning to look at a larger set, or all 50 lines?
Yeah, I think for now, the short term, we'll definitely look at the small subset of lines. Where Raju at Agri-Food Canada is growing different canola plants for us. So I think we're focusing in on just a subset and really looking at to see if there's a difference in the flow of ABA in those different plants first, and then we can maybe venture off into other broader categories. I think it would be really interesting to have different transgenic plants that say known genes that are involved in transporting ABA or maybe metabolizing ABA, and then looking at some of those individuals and see how the ABA flow changes, as well as the metabolism. I think that's also an interesting thing. And one of the things we're really excited about is Sue Abrams, who most people know is has 300 some papers on ABA analogs. And so we can start looking at how those analogs affect transport of ABA in plants. And that's one of the sort of big strengths of PET in the biomedical community, is helping drug design. So I think we're really excited to see how we can use PET to design agrochemicals, especially when they're based on the ABA scaffold.
Okay, that was another question I had actually. Have you actually started to look at some of those analogs that can modulate different responses to stress? I guess abiotic stress chiefly, maybe a main interest. But has that work begun, or are you still just working with standard ABA?
I think we're just working with standard ABA for now, and there's a few more experiments that we want to do. For example, we tried to extract the sap from some of the plants, and the funny thing is that the timing of these experiments it's a pretty big. You have to be very organized to get the plants grown to a certain size, because the PET scanner we're using, for example, it's for rodents. So we've had a few issues where cyclotron was shut down and all our plants got too big. So we had to restart and that sort of thing. So yeah, we're focusing in on those right now, and we'll be looking at the analogs hopefully very shortly, once we characterize some of the other experiments that we're wanting to do in the plants that we have.
Okay, and the focus is primarily on ABA, but I guess there are other plant hormones that might be of interest. Has there been any kind of thought about looking at other hormones?
I mean, to synthesize a PET tracer, so say we got excited to develop a new PET tracer for a different hormone, the radiochemistry can take years to iron out, because it's so bloody challenging. You're handling nanograms of an isotope. So part per million contamination in your solvents can have an effect on the radiochemistry. We have things like, radio tracer synthesis that work wonderfully in Vancouver, but fail in Saskatoon because we have different cyclotron systems, and the contaminants in the actual systems can affect the radiochemistry. That being said, there's nothing stopping us from, say, treating the plant with a different hormone, non-radioactive, and seeing if that has an influence on ABA transport or metabolism or other types of things. So to answer your question, are we going to make a PET tracer for other hormones? No plans in the future. Could collaborators such as EG and Raju come up with interesting biology experiments to do that could utilize the fluoro ABA? That's a good possibility and something to consider for sure.
There is a follow-up question from Nihar. So has the binding kinetics of the radio probe been checked with different ABA receptors? So there's this range of receptors in the plants. Or is there a plan to actually sort of look at this study downstream?
Yeah, I think we would start getting into the details downstream, and looking at the specific mechanisms of transport and transport proteins and so on and so forth. And that's where, once we understand if Sue has certain antagonists of a protein, can we block the transport of the radio tracer? And that would provide in vivo target engagement of specific proteins, or use a transgenic plant that lacks that protein. So that's definitely something downstream. And me, myself, being a more of a biomedical researcher getting into the plant space, I'm learning more and more about plant biology and ABA in particular. So I think that's something that personally I would like to pursue as the project matures, for sure.
Right. One last question I have is the PET detector, the plant one, which is now available, and you're getting those nice 3D images, which you showed. Is it just one of those? Is there a plan to develop more, or actually at some point look at producing one that could actually be potentially commercialized in future? Because I imagine there might be a niche market here.
Yeah, for sure. And that's the Aram and Zesis from the University of Regina have custom made this BioPET. And so do they have plans to commercialize it? Probably not. I mean, there's even a lot of, despite the popularity of PET in biomedical research, there's even a lot of companies have stopped selling microPET. There's a few that are really good at it. So perhaps they might have plans to do that. And a lot of plant researchers, if you do have a local cyclotron, we're using the microPET animal scanner, which can fit 2 rats in it, 4 mice. And that limits you to small plants, but, it's working fairly well for us. So I think long-term, the BioPET, as Aram and Zesis call it, will allow us to do those experiments in larger plants. And I think that's the real strength of their instrument. But since it's an experimental instrument, we're going to have a lot of validation to do in terms of making sure that, the actual radio tracer images represent actual radio tracer flow. And so we said we would help them along with that when they're ready.
Okay, thanks. Yeah, I guess the standard PET scanners, the ones that you showed for the smaller animals, are actually just like a cylinder which the animals are in. Is that? So yeah, I imagine you can get smaller plant tissues in there, but anything larger, it becomes limiting.
Yeah, and that's why the coupling PET with autoradiography as well can be very helpful, because then you could, do a larger canola and then section it off, and we got a number of plates that we could do different leaves with, or the root system, and so on and so forth. So yeah, I think we're limited to small plants for the PET imaging, but if we couple it with other imaging techniques, then we can start looking at larger size plants and more variety. Yeah, because these plants are laying sideways in a little tube. We put them in a tube just so that the soil doesn't end up in the PET scanner. The facility won't want dirt in their micro PET. So, but yeah, it's like a little stage, and usually you have 4 little mice there who are put to sleep with anesthesia. So, yeah, that I don't know what the biological consequences of taking a plant and placing it on its side are, but it might have an effect. I don't know.
Okay. Well, thanks, Chris. Appreciate it.
Thanks, Andy.
So moving on to Venkat, and there's a question in the feed for you. It's actually from Carl Venkat, and it's I guess something I was also going to ask. But the Symvisio tool that you described, that you've been working on, you've developed, with I guess more and more we're generating multiple genomes, not just one genome, any one species, but we have multiple genomes, and we're interested in comparisons between them, this pan-genome aspect, and trying to work out what are the key differences between multiple available genomes. So essentially these are de novo genome assemblies. So I guess the question from Colin for me is how extendable is the system that you've developed to that kind of context, that pan-genome analysis?
Thanks for the question, Carl, and the follow-up, Andy. To answer that, in theory, yes, it can be adapted to pan-genome datasets. We've already done this for a couple of small-scale pan-genome datasets, but are yet to extend them to really large-scale datasets beyond 10 or say 20 plus genomes at once. We have been sort of recently working on certain adaptations where collinearity is visualized across 4 lentil genomes through small multiple style visualization, where each visual is kind of situated in an n cross n matrix. We've also experimented with conservation between subgenomes of wheat across multiple lines. So, and sort of tree style graphs where conservation can be traced across several levels. But so far, our research has sort of been largely limited by our access to diverse pan-genome datasets. So if any researchers out there watching this today do have access to say pan-genome collinearity or Centeny datasets between multiple genomes and are interested in exploring them, we'd be really happy to visualize it for you. Realistically, it's mostly about extending the systems across multiple dimensions based on what the researcher is actually trying to look at. And in terms of visualization research, pan-genome visualization is a really novel field because nobody has really explored it much. So we are super interested in extending our work. And to sort of answer the question again, yes, Symvisio can definitely be used for pan-genome analysis, and we'd be happy to share examples where we have done that in case anyone's interested.
Yeah, I guess in theory, the system could be adapted to other types of omics data. I mean, you've got transcript data already incorporated, but things like epigenetic data as well. So methylation profiles could easily be included. I would imagine could easily be included and represented in the platform.
Yeah, for sure. Recently, we've also extended the system to sort of visualize ancestral rearrangements, like Dr. Lingling's research. We've actually taken that data set and then adapted Symvisio to sort of work with that. We've also worked with linkage group visualizations where you've just sort of sequenced a new genome and are trying to look at how similar it's to an existing genome. So we've also visualized that. So it all depends on the kind of data sets that you have and how the system needs to be adapted to that.
And then I've got another question. which kind of, I guess it sort of comes back to the operating principles for your lab. It's the interaction, the computer interaction laboratory. And I mean, one really pleasing aspect about the platform you developed, it's very easy on the eye, it's intuitive, it's nice to work with. So how much of the building process for the platform was built around kind of like your existing experiences or operating paradigm that you've developed, perhaps working in other completely unrelated disciplines, and how much were you reliant on the input from PIs in the PERT program that had genomic data?
So most of our requirements for the tool sort of came from PIs who've already been looking at existing data sets. And mostly what we have seen is most researchers kind of just use visualization tools in the end stage of their research cycle just to sort of publish them. And so we sort of wanted to solve this problem and actually move these visualization tools a couple of steps ahead in the research cycle, where they can actually use them to interact with the data sets and investigate different scenarios. And that's mostly what we have done. We sort of took existing visualization principles, things like, start at the large scale level and then drill down all the way to the gene level. And then we've also done multi-scale systems where you can look at the same visualization in different resolutions based on what you want to do. So it's sort of a mix between existing interaction principles and then trying to adapt them to genomic data sets in general.
Yeah, I guess there's kind of like a follow-up question which just come in here, basically noting close interactions between computer and genomic scientists as a key contribution to the visualization project. Yeah, which is definitely true. But in terms of how you carried out the process, do we using interviews, or what type of interactions did you deploy with the researchers to get the appropriate feedback and developing the prototypes?
So we usually start off with just the data set, and then we sketch around different visualizations based on the initial research question. And then once we have a base prototype up and running, we then do interviews with different research teams with sort of different diverse data sets, and then ask them if that answers the research question, and then come back with any sort of feedback that they've given, and then update the system based on that. And so far it's been really good, because most of our researchers have been really pleased with the end systems that we have developed, and we sort of are looking at exploring them beyond the original use case too.
Right. I guess it's important to note that SynVisio is just one tool that's available. The platform, there's actually a range of different tools that have been developed, I guess, over the course of the last few years. So certainly encourage people who are interested to check out the website, and I think you provided the link in your talk for that.
Yeah, Andy. So all of our tools have actually been hosted on genomevis.usask.ca. So please do visit that. And then there's detailed descriptions of what each tool does and how to access them. And again, all of our source code has been open source, so you can definitely just even adapt them into your own servers if you're interested.
Okay. Thanks, Venkat. Okay, so I have, I guess, a big question. I guess maybe it's a multi-part question, but it's to all the panelists, and hopefully you can provide some feedback to the question. It'd be good to get your thoughts on it. But basically, I want to focus on what you think is some of the big challenges, or perhaps opportunities, moving forward in a post-PERT world. I guess we're coming towards the end of the last year or two of the PERT program, but essentially we now have platforms that can generate huge amounts of data, multi-scale data, genomics, phenomics, etc. So what do we need to get the most out of these data sets? I guess that's one question. And where do you think things will be going, I guess, over the next decade? I guess the digital horizon. So coming back to the theme for the symposium. So I guess things to maybe think about. How much do we need to think about experimental designs? Do we need better experimental designs for the types of data that can be generated? do we need more input from experts in statistics? So for aiding the development of experimental designs. Do we need more data analytics at scale? So essentially computer resources to enable us to do large scale work. Do we need more intuitive data and result viewing software platforms? I guess this is an interdisciplinary program. But do we need to get even more interdisciplinary? Or is there a limit to how much we should be thinking about? Anyway, some thoughts from the speakers would be useful. And perhaps we'll start again with Cranos, and we'll just go around the table again and see how things go.
All right, thank you, Andy, for that question. I think that's something that we all have probably given tremendous thought to. I think some of the things that we really see in my lab and the groups that I work with as some of the most serious challenges is how to organize and collate the heterogeneous data that's being generated, not only within individual groups, but across different groups and across different research questions. There's obviously tremendous potential synergy in terms of decision-making aspects that can result from the data that's generated. But I think that because they're so disparate, and in some cases, organized in a way that does not lend itself to translation or transition into data analytics, it becomes extremely problematic. So I think looking at novel tools for being able to manage various types of agricultural and plant sciences data, I think is going to be a key step and a key facilitator in terms of being able to make actionable decisions and interpretations from the data that's generated. The other aspect is obviously going to be on the analytics side. And to some extent, we oftentimes look at this from the computational standpoint in terms of being able to perform operations at scale. And for me, although that is necessary, I think that another component that really needs to be considered is how these particular types of approaches can be used with the data that's generated. Unfortunately, all data are not the same. And their origins and the way that they're generated does not necessarily make them amenable to all types of analysis. So, for example, with microbiome data, its compositional nature really prohibits, aspects of using traditional correlations because they, in general, may not make a lot of sense because of how that data was generated. So really looking at the types of modifications that are going to be needed for these different types of analytics tools that are applied in these other disciplines and in these other areas, and how they should be potentially modified to be able to accommodate and be used properly in heterogeneous plant sciences or agricultural data, I think becomes necessary. So thus research in those areas, research in the potential changes that are needed are going to be effective. And then the other aspect that we've always kind of looked at is delivery. And this kind of comes down to the either basic science or translational nature of the work that's being done. If it's, for example, directly for specific stakeholders, being able to translate the machine learning approaches, the modeling approaches, et cetera, to the people that really need it in terms of making decisions on the outputs of these algorithms is going to be key. And translating this information to a scientist looks different than translating it to a grower or translating it to a packer. So being able to really focus on how we're going to be relaying the decisions that are being made from these analytics to the proper stakeholders, then really not only helps to exacerbate the potential effectiveness of the work that's being done, but also serves as a feedback in terms of then the interpretation and then what could be done differently or what modification needs to be made in terms of either data generated or algorithms that are designed.
Yeah, I think that's a really important point, Cranos, that delivery of the outputs, the outcomes from the work that you do. It really depends on the context with the different stakeholders that tailoring things in a way that makes it interpretable to different stakeholders is really important. And I think certainly moving forward for the PET, that's an aspect which has to be more developed, especially when looking towards representing things to producer groups or producers. So then you can really hope to convey the significance of the work and how it impacts people growing the crops in the field. The data management side is one thing, not just data management, but how you do it and what you're trying to represent as key in metadata. So the other information associated with data sets is really key as well. I guess one thing that we had tried to do is have a focus on particular germplasm resources of the NAM population. The canola NAM population has been mentioned a few times in the talks here, and that's actually gives you the ability to have some consistency in terms of a resource which is used, and then a lot of data is actually generated from it. So you end up with a range of different data sets that you can then start to perhaps draw significant analyses from those data sets for that same population, genetically identical population. The nice thing about it is that you can grow the population in different environments, different locations. Essentially, it's the same genetic composition for the population, no matter what you do with it, whether it's in the field or in a controlled environment. Mik, do you have any thoughts?
Yeah. Sorry, I was just listening. I think I agree with all those points from Cranos. The delivery is also, I can see from a modeling perspective why that's pretty important, because we want to make, say, models that will be useful in some way to help advance research or to help growers in some, design or predicting their yields or yield potentials or their crop growth. I think I would add though that yes, again, for modeling, we'd want to help to advance just our basic understanding of how plants grow. So can I make a model that incorporates all the different known mechanisms or mechanisms that we don't know yet, and will that reflect reality? I think it's before delivering tools that can be used to predict how crops grow, we need to have a pretty good understanding of what controls their growth. So, for example, the ABA treatment studies, I mean, they're involved. I believe it's involved in the control of branching, and that is a very huge topic, with many groups around the world studying just control of branching. Well, not just control of branching, but lots of groups study control of branching. And if we are able in the future to make a model that can incorporate the kind of most important aspects of the control of branching, I mean, that will help our basic understanding of how it actually happens inside the plant, and then that might lead to more kind of predictive applications.
Okay, thanks. I'm just keeping an eye on the clock. We'll just have 7 minutes left in the session, so I'll move on to Chris and see if you have any thoughts.
Sure, just really quickly, and again, I'm new to the crop sciences, so maybe I'm not reflecting reality here, but it sounds, what the other two speakers had said, sounds bang on. So I'll address the collaborations with other scientists. One of the things I've noticed is that there's a lack of chemical tools to study enzyme and protein function in vivo in a plant. So there seems to be a need for fluorescent probes, radio tracers, enzyme inhibitors. There's a lot of reliance based just on genomics. And I can tell you, in the other enzymes I study in biomedicine, expression does not equal activity. So that's a big problem, is that we've got cancers where expression is the same, yet a dramatic increase in the enzymes that create a permissive environment for tumors to invade, cancer to invade, goes up. And same as another enzyme in Parkinson's. So I see that there's really a lot of need for sort of chemical tool development for measuring enzyme activity in a living plant, inhibiting an enzyme, or fluorescent probes, for example, that could be used to profile enzyme activity in a plant, run an SDS page to profile those and get a hold of the activity data. So, anecdotally, that's where I see there's a big need. And then just really quickly, secondly, the image processing in PET imaging definitely needs to be improved. For example, I'm not sure about the other areas, but that's another area that I think could really help increase the impact of radionuclide-based imaging is the image processing implants, which is not easy as in mice or humans.
So, yeah, the first part that you talked about, Chris, there sort of reflects perhaps on where we need a greater interdisciplinary connection, right? Where that a better ability to take on board the chemical, the biochemical aspects into future programs would be really potentially quite valuable. Right, and finally, Venkat, just with respect to your perhaps with a focus on the viewing software platforms, do you have any comments?
Sure, Andy. I know this has already been touched upon a couple of times today, but I sort of feel that a central data repository for all the PERC researchers would be great, because one of our biggest roadblocks so far has been in getting data across research teams, and this is not helped by the fact that genomic data is really, really big. So transporting it between research teams is even more difficult than usual. Additionally, such a central database would be useful for us developers, because we could then link them directly to our system, so researchers don't have to jump through extra hoops to access them. And it would kind of increase the general audience beyond the direct research teams we work with. And of course, I know that researchers can be reluctant to share their research data pre-publication, but I'm sure a sort of permission system can be built into any data repository to limit access to who sees it and who doesn't. So yeah, a central database would be great if we can have that.
Right. I guess there is a such a resource effectively in development, or has been developed. It's a ongoing process in terms of development, and there'll be, I think, a description of that. I don't know if it's this afternoon or tomorrow, but I think ensuring awareness of the resource and how it can be utilised optimally for the PERC initiative has got to be the next stage of implementation of that platform. Okay, I think I'm going to have to end the session now because we are about to run out of time, just a couple of minutes left. So I really want to give my thanks to all the speakers for the session, the time in providing the talks, and the time in the question and answer, and of course the audience for joining us today. It's really appreciated, and hopefully you'll enjoy the rest of the symposium. The next session will begin at 1 p.m. Mountain Time, followed by a debut of a special recorded panel with Nutrien on the carbon opportunity in agriculture, and that will be at 3 p.m. Mountain Time. Also, if you haven't already, please take some time to check out the 6th annual poster session, which can be found on the homepage of the symposium, and please vote for your favourite poster. We have a People's Choice competition. And with that, I'd like to thank you again, and look forward to seeing you this afternoon at 1 p.m. Thank you, and bye.